blood samples. And what you are going to come up at the end of your analytical process is this sort here. I don't know if you see the cursor of my mouse, but you can see it moving. You're going to come up with an abundance matrix with in rows the genes and in columns the samples. So it's a matrix of read counts, integers. On the other hand, with DNA methylation, what you will come up with usually is a proportion of the level of methylation for a given regulatory site. And usually you have this common pattern that is commonly observed. The more methylated you are for your regulatory sites, the more the expression of your corresponding gene expression is altered or is decreased. However, in my talk, I will mostly focus on the decoration algorithms that rely on bulk transcriptomics for better characterizing biological profiles, heterogeneous biological profiles. But just for your own knowledge, know that there are many decoration algorithms that also rely on DNA methylation, and there are even some really recent approaches, I think 2 or 3 decoration algorithms, that try to integrate both information. Just a side note, if you don't understand some of the concepts, I don't know how exactly you can iterate myself, but do not hesitate to interrupt. Okay, so when you are doing standard bulk transcriptomic analysis, you're going to go through all these stages. Usually you have a stage of data management where you come up with an expression set. Then you have a data quantification stage, in which the key stages are the removal of the background expression, so the genes that reveal unexpressed, the discarding of low quality samples, for example, the ones that poorly overlap with other samples, normalization, and transformations. Normalization is usually performed to ensure that your samples are comparable with each other in your analytical framework. So once you're doing all this pre-processing analysis, you're going to go through a variety of quality control and visualizations to ensure that your data pre-processing and data characterization was performed correctly. And so usually what you want is that your expression is comparable across samples, that you correct for batch effects without losing the biological signal. So usually you want to perfectly separate, discriminate controls from disease case, for example. And once you have done all of this, you will go through and undergo a variety of downstream analysis. And usually the most popular, the most common one is differential expression analysis, in which you are going to quantify variations of gene expression between 2 conditions. Usually, so you are going to consider one for which the change of mean expression is the most significant. Actually, the first stage or the first step of my PhD thesis consists of deriving a whole automating transcriptomic pipeline and to use the outputs of this transcriptomic pipeline to stratify the patients suffering from Sjogren's disease. So Sjogren's disease is a disease it's an autoimmune disease that mostly affects women, and which is not really life-threatening, but it's really debilitating in your everyday life.
Doctor, I'm sorry to interrupt you. I think your presentation has frozen because the slides are not changing.
Okay. I'm trying to reshare. Oops, sorry for that.
It's okay.
So normally you should see a heat map from now on.
Yeah.
Okay, perfect. Please feel free to interrupt if there's any network issue or if there are some questions occurring in the channel.
Of course.
Okay. So this is what I've been performing. And so once I have derived the clusters of all my patients, I'm going to try to identify the markers, so here the transcripts, that explain the assignment of an individual to a given cluster or to another. For example, here it's a really simplified example. I observed the significant change of gene expression for a given gene between the healthy state condition and the disease state condition. So what happened there? In most cases, biologists will assume that there has been an activation of the pathways of the cell population, of the pool of cell populations natively present in your sample. But there's another scenario to consider, which occurs quite often actually, which is maybe as equally feasible and plausible as scenario A, which is a change of cell composition with infiltration and the migration of a new cell population. And this cell population, here characterized with the pentagonic shape, which release this gene in higher amounts of gene expression. And actually, when we rerun the analysis and we try to characterize the patient cluster that have been automatically retrieved from data-driven approaches in Sjogren's disease, we noticed that most of the discrepancies observed across clusters could be characterized or were driven by changes of cell composition rather than activation of pathways, of cellular pathways, of transcriptomic pathways, sorry. Okay, so this is where we are going to need other approaches, either numerical or technical ones, to better understand the causal variations and the causal mechanisms that lead to altered mechanisms across cohorts. So in the first case, you may rely on technical approaches. So at first, of course, we had only bulk omics, which mixes everything, which is represented here with the smoothies. And after some time, we came up with single cell. So with single cell, we both have the individual information for each of the single cell, for each of the cells within your mixture, and the characteristics of each of the cells. And we even go further, maybe 2 or 3 years.
I'm sorry again. The slides are not changing. It's still on the Sjogren's disease.
Oh, okay. Okay.
You want to share it, and you tell me.
Yeah. Maybe we can switch. Wait, can we switch? Maybe if you share the screen, this issue, this technical issue, will be mitigated. No, I don't know.
Okay.
And I can say, yeah, you can go to the other slide.
Can you change? See if you can change the slides now.
Okay, so you change the screen. Is there any shortcut to do so?
There is like a an arrow on the end of the page. If you bring your cursor down.
On the screen that you're currently sharing. Huh?
Yeah. You can't see it, huh?
No, I can't see any arrow, and I can't. Yeah. Sorry.
It's okay. I'll do it. You just tell me when to change them.
Mm-hmm. So just go to the slide that I was presenting. Yeah, keep on. And keep on. Yup. Yeah, perfect. Yeah, perfect. Stop here. No, yeah, you can go to the other slide, actually. Yeah, perfect. So the main issue in my case is that at that time, actually, the pharmaceutical company Les Laboratoires Servier didn't have the technical and computational resources to run single cell or spatial transcriptomics. Secondly, in some cases, actually in most cases, you have legacy datasets. So you don't have access anymore to the raw biological materials. In other words, to the human samples or to the humans included in the cohort. So what do you do with your legacy historical bulk transcriptomic analysis? Finally, with both single cell and spatial transcriptomics, these technologies are quite hindered by some specific technical artifacts that are specific to the methods. For example, with second cells, I think up to quite recently, we were not able to capture neutrophiles, which usually is like the most prevalent immune cell type in a given blood sample. So that's why we may need to resort to other kinds of approaches. And you may, I guess, pass. please. We are going to rely on computational numerical approaches. So there are two kinds. So there's like a whole family of deconvolution approaches that have been developed so far, but usually they are discriminated and separated between partial deconvolution approaches and complete deconvolution approaches. In partial deconvolution approaches, you're going to provide some prior knowledge. So in any case, you're going to provide, of course, the bulk transcriptomic profile that you want to deconvolve, but you're going also to provide either the cell-specific expression profiles or the individual sample-specific expression as computed, for example, with FACS. On the other hand, with complete deconvolution approaches, you only have access to the bulk transcriptomic material and you try to simultaneously infer the cellular ratios and the cell-specific expression profiles. And it turned out, pass.
Doctor, can I ask you something? I'm sorry.
Yeah.
Because the 15 or so of the slides weren't shown when you were talking. Can I go through them once and you just give a summary so they can get to know the stuff better because they haven't seen them?
Sorry?
The slides were freezed and people couldn't see them till the last slide. So can I go through them once more and you talk about them for 1 or 2 sentences? So
okay. So from which slide does it start freezing?
It was at the first.
Really? Oh,
yeah.
Okay. So really, there's an issue. Is it the RSG platform that is not working actually?
No, it was ours. It's okay.
Okay. Okay. So you can pause this one. So as I said really briefly, the overarching idea of computational biology is to try to identify the variations of biological compounds that lead or that switch from a balanced system to an altered one. Pass. And the compounds that are usually considered for deconvolution approaches, and most actually omics approaches, is bulk transcriptomics and DNA methylation. Pass. So usually, and this is the case in my deconvolution algorithm, I will mostly delve into bulk transcriptomics. In other words, bulk transcriptomic, it quantifies the level of expression of RNA molecules in a soup. And is going to return this kind of matrices, of abundance matrices, with the gene expression in rows and call names, samples in call names. And you're going to come up with this matrix of root counts. So here, this is just the traditional workflow for bulk transcriptomic analysis. And I would say the two main maybe key points to keep in mind in this slide is that we are going to go through a variety of pre-processing operations to guarantee first that your samples are comparable with each other across batches, and second, that the low quality samples or the unexpressed genes are discarded because they are not relevant for your downstream analysis. Pass. So I did apply this automatic bulk transcriptomic profile to better characterize the Sjogren's immune disease. More specifically, we came up and succeeded to identify 4 endotypes within a cohort of patients suffering from transcriptomic profiles, and we identified 4 subgroups of them, which seem to exhibit distinct biological molecular processes. Pass. So, of course, when you came up with these 4 clusters, what you want to infer and what you want to go further, to go subsequently, is to determine the most relevant biomarkers that can explain the change from one state to another. So this is the general overarching of differential analysis. And one of the premises when you observe a significant change of expression is that you have an induction of the cell population natively present in your sample. But pass. Another equal hypothesis is to consider that you have a change of cell composition with the infiltration of a new cell population. And this is this new population that releases in much higher amounts the gene expression that has been changed across conditions. Pass. And actually, this is what certainly occurred, and this is what certainly characterized best the clusters. So not changes, not native changes of expression, but rather changes of cell composition. Pass. Okay. Pass. Yep, here it is. So the problem is that with just bulk transcriptomic, you can't settle whether it's hypothesis A, so native induction of the pathways within your cell population, or scenario B, a change of cell composition. So you will need to rely on other approaches. One way of doing so is to rely on technical approaches, like, for example, single cell or spatial transcriptomics. With single cell or spatial transcriptomics, usually you will have information at the cell level, at the single cell level, and not at the whole bulk level. And you will also have the specific expression for your given cell type. But, pass. There are plenty of studies where you do not have access because maybe you do not have any access anymore. You don't have access anymore to the raw material. So, namely, the main humans that were included, patients, that were included in the cohort, or maybe it's just a legacy issue. It's a legacy issue, so you can't really access it, and you only have access to bulk transcriptomic, or so on, etc. So you don't have this information. So the single cell or the spatial transcriptomic information. Another issue with single cell, and maybe it's a bit it's less critical for spatial transcriptomic, but that's still an issue, is that these two approaches, they come up with much more challenging technical artifacts and technical issues. For example, in the case of single cell, we are usually not able to capture neutrophiles, while neutrophiles is the cell type which is certainly the most frequent in a given blood sample. So what to do when you don't have the money or the resources to perform single cell or spatial transcriptomics? Pass. You're going to rely on computational approaches. So there are plenty of deconvolution algorithms that have been developed so far, but I would say, the most common are called or termed partial deconvolution algorithms. Why that? Because you're going to provide and to help your deconvolution algorithm by providing prior knowledge. So you can either provide cellular ratios, as quantified by facts, for example, or cell-specific expression profiles. And in that latter case, you want to infer the compositional nature of your sample. Pass. This is the case, or this is the purpose, or this is the category in which DecoVart falls in. Pass. Another thing to consider beyond the application of your deconvolution algorithm is the level of granularity you want to consider, the level of resolution. Indeed, there are deconvolution algorithms that are really at a low tier level and that work at the tissue level. And usually this kind of algorithm, what they want to infer is the level of contamination in a sample. And to be even more specific, the level of tumoral proportion of tumoral cells, in a tumoral sample. Because usually, especially in solid tumors, when you're going to retrieve and extract your tumor using, for example, a biopsy, you will notice an intertwined blend of healthy and tumoral tissues. And the issue is that you don't have directly access to the characteristic of the tumoral tissue expression, because usually it's so specific to a given tumor, to a given patient, to a given set of mutations. At a higher level of granularity, you can infer cell population. So this is the most typical approach, the most common approach for most deconvolution, most common objective for most deconvolution approaches. And finally, you could even go finer by considering the cell stages or cell states within a given cell population. Indeed, you're not going to witness the same characteristic or the same profiles, whether your cell is in its phase of proliferating or growing or replicating. Pass. Okay. So let's go a bit further into the technical aspects and into the maths. So here with the matrix while I just represented, I'm just showing the bulk expression profile for J genes that I carefully selected and for N individuals. So it's the total bulk transcriptomic expression that I'm observing. Secondly, pass. I'm going to provide purified transcriptomic profiles as noted by X, matrix X, for my J cell populations, J genes, and G cell populations. And usually we are only considering the mean expression profile. And with these two inputs, pass, we are going with numerical computational approaches to infer the cellular ratios for each of my sample. So usually this is performed independently for each of my transcriptomic sample. Pass. Secondly, most of the decorrelation algorithms, which makes quite biological sense actually, they will consider a linear assumption between the bulk transcriptomic profile and the cell-specific expression profiles. So to be more specific, you are going to assume, or they're going to assume, that the bulk transcriptomic profile can be reconstructing by summing the individual cell-specific expressions weighted by the relative abundances of your cell population in your sample. So this is depicted by the first equation, the first matricial equation. Secondly, you want your cellular ratios to adhere to the unit simplex constraint. To be more precise, we are talking about ratios. So if we are talking about ratios, these ratios, they must be included between 0 and 1, relative ratios. Secondly, I'm considering that all my cell types possible, available in my sample, are characterized by the provided cell-specific expression profile, so by matrix X. In other words, I'm going to consider that the sum of my cellular ratios is equal to 1, but there's no unknown cell tumoral content, there's no unknown cell content that may contribute to the global bulk transcriptomic profile. And so these inequalities are captured by these constraints are captured by the second set of inequalities, here illustrated in red. Pass. Okay. So as I said, the purpose of the cohort is to go further and to integrate the gene regulatory information within my generative framework. Go further. Pass. So just a small reminder for you, here we are considering the mean expression profile. So it means that we are considering that the expression for a given cell population is static, is fixed, is determined, which doesn't really make sense in biology because we know that there are some variations due to technical artifacts, due to just stochastic biological processes, due to maybe different phenotypical characteristics, or like variations across individuals or disease, and so on. So the idea, pass, is to consider that instead of a fixed distribution, we are going to consider a stochastic distribution of my gene expression. And even further, we are going to consider the interactions between the genes, as captured here by a network. So to be even more precise, in this network, the genes or the transcripts are represented here by the vertices, so the round shapes, and the interactions between them by the edges. Pass. Okay, so now the question is, how can we integrate this gene regulatory information within the pipeline, within a standard decomposition pipeline? So the first stage of any standard decomposition pipeline is to collect an enough compendium of datasets to characterize my cell-specific expression profiles. Pass. So yeah, I won't really go further in this task because it's mostly data management, but I will just add two innovations that I partly contributed. The first contribution is that I consider cell ontologies to homogenize the nomenclature of my cell population using package ontomproc. Pass. And the second innovation is that I consider statistical approaches, and more precisely parametric mixture models, to discard and to retrieve automatically the signal from my transcriptomic expressions. So I'm going to remove the background noise. Pass. Pass. Okay. So now that I have collected all these datasets, that I have cleaned them, the next stage is, of course, to infer the characteristics, the features of each of my cell profiles. Pass. And remember, I told you that I'm not only interested in the mean expression profile, but also in the gene regulatory networks. So there are plenty of approaches that have been developed to depict the gene regulatory information or to networks and the interplayed connections between genes. One of these approaches, and I will tell you more about this, and for the reason for that is the multivariate Gaussian distribution. Why that? The multivariate Gaussian distribution exhibits one parameter, which is the covariance, or more precisely, the inverse of it, which is the precision matrix, that enables to directly capture and encode pairwise interactions between two transcripts, conditioned on all over. So you're going to remove spurious correlations across genes. To be even more precise, if you have a non-null diagonal term in your off term, so for example here between CD19 and CD3, it means that you have a direct connection between these two genes. Pass. And so here is just another representation. You're going to infer this network for each of your cell populations. One of the major issues is that usually I have much more genes considered in my framework, like for example 2000, with respect to the number of samples I have access to. For example, to characterize dendritic cells at the population level, I could only be able to retrieve maybe 4 or 5 samples. Pass. But this is not that much, I would say, an issue for Gaussian mixture model-based approaches, because there's a variety of approaches, statistical approaches, that have been developed to overcome this curse of dimensionality. And certainly one of the most popular is called the graphical lasso. So the graphical lasso mixes a standard likelihood, so here represented in red, with a penalty regularization term that would penalize for spurious interactions between genes. So only the most significant interactions would be considered. Another way of considering the graphical lasso is a sort of multi-dimensional extension of the standard lasso regularization framework. Pass. Another idea to overcome the curse of dimensionality is to add single-cell RNA-seq profiles, because there are plenty of network approaches that have been developed to infer gene regulatory networks and the single-cell population. In particular, there has been a benchmark that has been more or less recently published, so term B-line. I think there are more recent benchmarks that have been developed so far, but what was exhibited in B-line is that the two best performing approaches were GNIS3 and GRN Boost 2, and they overall exhibit really great stability in inferring these gene regulatory networks, and they are not that much sensitive to the dropouts. So the dropouts is all these zeros that we observed in single cell and RNA-seq expression profiles. However, unfortunately, some studies tend to show that the gene regulatory network that were inferred with single cell profiles could not be mapped or were quite diverging compared with the ones inferred from bulk transcriptomics. So final first strategy to overcome the curse of dimensionality, integrate prior information, or in other words, bottom-up approach. Pass. So more precisely, there's like a whole compendium of databases and resources like OmniPath or Decouple Air that directly provides these interactions between genes. And so I would say the objective here is to minimize the really high-dimensional space, high-dimensional input space, so the gene's transcript space, by projecting it to a much reduced one at the pathway level, and the pathway level will be characterized by the transcription factor activity score. Okay, so these are three ideas. I did only integrate the first one for the moment so far. Okay, so here I'm now considering that I've been able to retrieve the networks for each of my cell population. But still, I usually came up with too many markers. And there are plenty of benchmarks, or there are 2 or 3 benchmarks that definitely show that the best performances were reached when you are trying to minimize the subset of markers required for characterizing your cell population. So how to determine the minimal set of markers to characterize a given cell population? You're going to do it in 2 stages. The first stage, you're going to identify the subset of markers specific of a given cell population with respect to all others. Secondly, remember that we are considering not only the mean expression profiles, but also the network expression. And so this is something that I would like to incorporate when selecting my markers. And one way of doing so is to rely on general class of family, which is called differential network approaches. Pass. So for example, indeed belongs to that category and is going to compute a sort of activity score that computes, or that is sort of a proxy of the level of magnitude and importance of a gene for characterizing a given cell population. And this activity score is computing by both the fold change, so the variation of mean expression between cell type, a given cell type with respect to all others, but is going to compute also and to integrate network rewiring, so network rewirings, so changes of networks and neighborhoods across cell populations. And it turned out actually that there are some cell types that are really closely related, so they will exhibit the same expression profile, on average, but the connections between the genes will be different from one stage to the other. So this is really something that we would like to capture when you run this kind of analysis. Pass. Again, another approach, bottom-up, mechanistic-based, is to rely on the known pathway information and to retrieve the minimal subset of pathways and activation factors that could have led to the altered mechanism, or that could have led to the specific phenotype of a given cell type. For example, Carnival and Cornetto did implement this across biological conditions. Pass. Okay, so this was the first stage. But now you want to even refine, to refine even further your cell-specific expression profiles by considering not only one cell population versus all others, but all cell population against all others. So for this, you have 2 potential solutions. One way of doing so is to try to integrate different metrics and to optimize for different metrics simultaneously. This is something, for example, that has been done by AutoGene-S. And in AutoGene-S, you try to minimize the correlation between your cell profiles, as computing by Pearson correlation score, and to maximize the centroids, so the mean expression profiles, between your cell populations. Pass. Another way, and since we are talking about statistical distribution and we are modeling the generative model just using a statistical model, would be to consider metrics that have been developed to characterize the dissimilarities and distances between statistical distributions. And certainly one of the most popular is the overlap. And I really appreciate this overlap metric because it's really straightforward to be understood, both geometrically. So the overlap here, for example, I consider 2 distinct separations would be the area under the curve. Or if you want to interpret it probabilistically, is the total probability of misassigning a given expression profile to a given cell population, even when you know the true input parameters, even when you know the parameters, so namely the generatory networks, the covariance precision matrices, and the mean expression profiles. And so something that we would like to optimize is to reduce the overlap between your cell expression profiles. Pass. Okay, so now I will assume that I've been able to really characterize the generatory networks for each of my cell population, and that I've been able to retrieve the minimal subset of biomarkers for characterizing any of my cell populations. And that I want to incorporate this information now in the covert. Pass. Pass. Okay. So here I just represented with this sort of flowchart the families, the two main families that have been developed so far to infer the cellular ratios in samples. And you can classify them into regression-based and probabilistic-based. So in my case, since we are relying on statistical distributions, it's of course probabilistic-based. And to be more precise, in the probabilistic framework, you're going to try to model the distribution of your bulk expression profile, conditioned to the parameters that you want to infer, so namely the cell-specific expression profiles and the cellular ratios. To be even more precise and specific, you can either consider discrete distributions, so you start at the count level, or continuous distributions. And actually, since we are considering multivariate Gaussian distributions, which are continuous, the covert falls under that category. So far, up to my literature review, I was only able to come up with 2 or 3 deconvolutional algorithms that consider this continuous approach. Pass. So here, this is the DAG, so a graphical representation of the model of a standard linear model. And one thing that directly catches the beholder's eye is that the mean expression profiles, here characterized by the squared box, is determined and fixed. And the only discrepancy that is observed, the similarity which is observed between the bulk transcriptomic profile and the one that is reconstructing, summing the individual cell-specific contributions, only proceeds from the addition of a technical noise artifact to the bulk transcriptomic profile. So sort of error, technical measurement error. Pass. On the other hand, in the generative model of decovert, we are not considering anymore, or we are going to relax this determined fixed assumption of a given cell-specific profile, and instead we are considering that it proceeds from a multivariate Gaussian distribution. However, I'm considering still a strong premise here. I'm considering, of course, the interactions between the genes within a cell population, but, and as is characterized by the dashed square here, the dashed box surrounding the X profiles, I'm assuming that my cell profiles are independent with each other. So within a given cell profile, your genes can interact with each other, but your genes can't interact, or your cell-specific profiles can't interact across cell types. So yeah, this is a strong premise. Secondly, another premise that I'm going to consider, but that could be quite easily relaxed, is that the stochasticity or the variability and the discrepancy observed between the bulk transcriptomic profiles and the ones that are reconstructed within my generative model only proceed from the stochastic intrinsic stochasticity of my cell-specific expression profiles. And now you are going to understand better why I'm choosing multivariate Gaussian distribution. That's the third reason, beyond sparsity and the straightforwardness to represent networks, generatory networks. The third reason is that it turns out that a convolution, so in other words, the sum of independent random variables following a multivariate Gaussian distribution is identifiable also to a multivariate Gaussian distribution. And in other words, it's an explicit form. It turns out that for most, this direct relationship, this straightforward relationship, does not hold for most probability distribution, which is really something specific to the Gaussian distribution. Pass. So the good thing, if you have an explicit form of the log likelihood of your distribution, is that you may derive an explicit form of the gradient and deviation, and I was able to do so. However, quickly, another challenge arrives. I was not able to derive an explicit form of the mapping of the gradient, or in other words, how can I retrieve the values, so the scalar ratios, for which my gradient cancels out. In other words, the values for which we reach a maximum, because this is the purpose of maximum likelihood estimation, is to retrieve the ratios or the parameters that maximize your quantity of interest, because it's the most likely one. But that's not really an issue, because there are plenty of numerical optimization approaches, ranging from first order to second descent order approaches, that enable numerically to retrieve, using iterative runs, to retrieve these cellular ratios, these parameters of interest. So let's pass. So really, just for your own culture, you can see that the explicit formula of deviation is, I would say, quite esoteric and challenging, but it was not that hard using matrix workbook notation. So it's just for your own knowledge, if I can say that. So pass. Another challenge, statistical challenge that arises is that, remember, that I assume that my cellular ratios must adhere to the unit simplex constraint. In other words, that we have ratios and that they must sum to 1 and be strictly included between 0 and 1. And so if I'm not adding directly this constraint, this won't work. Maybe my ratios, they are going to go infinite, for example. So in order to enforce explicitly this constraint, I'm going to consider reparameterization of my framework using a sort of softmax transformation of my unconstrained free parameter, so rho here, to transform it and to map it back to the cellular ratios that are indeed constrained. And I was able to do so. Yep. Yeah, perfect. Okay, so as I said, that's still an ongoing work, and this is more or less where I stopped at the end of my PhD thesis. And unfortunately enough, I was not able to go much further, because there are other statistical challenges that I would like to solve. The first challenge, the third challenge, if I can say that, is to prove the uniqueness of the solution, because there's no theoretical guarantee that the solution of my maximum likelihood is unique. So maybe there are some local solutions that correspond to local maximum or saddle points, and maybe a gradient iterated approach will fall under that category. It seems to be not the case when just doing some numerical estimation approaches, but it should be proven theoretically. Fourth challenge. The fourth challenge that I quite overlooked at first, and after that I said, that's truly an issue, is that the cell-specific profiles at the individual sample are unobserved. In other words, what I have from other population level based profiles, what I have is the mean expression and the covariance matrix for each of my cell population. But the actual value in my sample, this is something that I do not have the access. So I need also, I should, in my generative model, try to incorporate and infer both my cellular ratios and my cell-specific expression profiles. And this is something that I need to do. But doing so, maybe I will come up with another issue, an identifiability issue. In other words, maybe there are an infinity of solutions, of pair of solutions, of ratios and specific profiles that can maximize my log likelihood. This is something to be integrated and tested for. Oh, pass. Finally, multivariate Gaussian approaches is really great because it really simplifies all the theoretical and analytical derivations. But actually, that's also really wrong. Indeed, when you come up with this abundance transcriptomic samples, expression matrices, usually it doesn't, when you plot them as density profiles, it doesn't really match Gaussian distribution. And usually you will need to log to transform your data sets before assuming it's normally distributed. But when you log transform your data, you're losing the fundamental linear assumption underlying your decoration framework. So namely that you can reconstruct your bulk expression profile by simply summing the individual contributions of your cell profiles within your sample. So how to or which other parametric candidate solution could be considered that could better fit the reality, that could better fit the compositional and abundance count nature of transcriptomic samples. I would say that certainly the most promising candidate is the Poisson log normal. First, with the Poisson part, you are going to properly characterize and to properly model the fact that your genes are clones, and integer values. Secondly, with the variance of your the mean parameter of the Poisson log normal distribution is going to be modeled as a multivariate Gaussian distribution. So in other words, you're more or less making the best of both worlds. Another great thing is that there's this package here termed PLN model, which is, I think, implemented on bioconductor, and which provides a variety of approaches to infer efficiently the parameters of a Poisson log normal distribution. Pass. As it turned out, from some recent benchmarks and for spatial transcriptomic decomposition, but I would say that there are really some similarities and sharp points between bulk transcriptomic decomposition and spatial decomposition approaches, is that, Poisson log normal seems to be relatively efficient for capturing the distribution and to be quite faithful to the nature of your distribution. However, in RCTD, so it has been developed by Raphael S. Izari from Boston, I think, or Massachusetts. It's one of the most popular spatial decomposition approach. It means for robust cell type decomposition. They consider indeed a Poisson log normal distribution, but they consider this Poisson log normal distribution only at the gene level. So in other words, they are not considering and accounting for the interactions occurring between the genes. So that's not exactly what I wanted to model. And to conclude my talk, and to showcase that it's indeed, or it seems to be relevant to integrate these pairwise interactions, I did just a very small toy example. So here, I'm considering two cell populations, characterized only by two genes. In all my modeling scenarios, the proportions are balanced. So it's the same proportions. It's the same centroid, so the same mean expression profiles, and it's the same gene-specific variance. The only thing that I'm playing with is the pairwise correlation, so one parameter, between my two genes for each of my cell population. Remember that decomposition algorithms, most of them, they assume static distributions. Or in other words, they will only consider the mean expression profiles. So for them, all the four scenarios that are being illustrated below, on the below part of the slide, are the same. However, when you're running the decomposition scripts, you can see that integrating and the level of correlation between the genes plays a strong impact on the performance of the decomposition algorithm. And as you may have guessed, of course, it's much easier to infer the proportions of cell populations when your cell types are strongly separated. So it's the bottom right corner, while it's much more complex and challenging to infer your ratios when your density distributions characterizing each of your cell populations are strongly overlapping, like it's the case in the top left corner. Just a point on the metric used, I just used the mean squared error estimate between the inferred cellular ratios and the ones that were used for running the simulation, so equivalence distributions. The other thing that need to be retrieved from this really simplified modeling framework is that Decovart seems to exhibit overall better performances compared to more traditional approaches like Decon RNA-seq, as you can see with the bluish colors. So the lower the mean square error estimate, the bluer the color, and the best performance overall. Okay, so let's conclude now. Pass. Okay, so in Decovart, two things, the two major innovations that I introduced in Decovart so far is first a sort of standardized workflow for annotating the cell types and tissues and diseases. and to remove the background noise. And secondly, critically, really the most relevant innovation is that I try to incorporate the gene regulatory network information at several stages of my deconvolution pipeline, both at the selection of the markers characterizing a given cell population and in the generative framework. And with just a really simplified modeling framework, we could see that, yes, indeed, integrating this gene regulatory information is critical for the performance of your deconvolution algorithm. However, there are plenty of avenues and tracks of improvement for decovert and other deconvolution approaches, actually. First thing, we would need to derive not only point-wise estimates of the cellular ratios, but also confidence intervals or lower metrics to quantify the level of uncertainty associated with the estimated cellular ratios. Now, this statistical section, it's often overlooked by most deconvolution algorithms. They will only return the cellular ratios, but you will never know the confidence you could bring to these results. The second thing that could be interesting to consider in decovert would be to be able to evaluate the impact of an unknown, for example, tumoral profile that has been not characterized or incorporated prior as a prior mean expression or gene regulatory profile in the generative model. And finally, if I want to be published, I guess, in a great journal, in a high impact journal, I would certainly need to tailor decovert not only for bulk transcriptomic, but also for spatial deconvolution. And yeah, that should not be too hard to do so, because as I said, one of the most popular approach is RCTD, which assume log normal distributions, but only at the gene level, so not accounting for the interaction of the genes, and it appears to be one of the best. Okay, so let's pass this slide. Pass. So I have more or less finished my presentation. Now I'm going to do, I would say, sort of networking and promotion of other initiatives that I find really cool in the field of deconvolution. The first thing is the OmniDeconv project that is currently, I think, supervised and managed by Francesca Finotello, currently located at Innsbruck. And the great thing is that they have developed plenty of packages to homogenize the whole process and to streamline the process of deconvolving your samples, relying on different deconvolution algorithms. Like, for example, I think in ImmuneDeconv, they implemented like 20 deconvolution algorithms to retrieve automatically the proportions derived from bulk transcriptomic profiles. In OmniDeconv, single cell. In Deconv Explorer, they provide, it's a shiny app to see the impact of selecting markers on the characterization of your cell types. SpaceDeconv, as you may guess, deconvolution for spatial transcriptomics. Deconv data, just a set of data sets. Simbu, for simulating realistic frameworks of deconvolution distributions. And finally, Deconv Bench, as you may guess and infer, is to compare the performance of deconvolution algorithms across scenarios. Okay, pass. Final slide, and it will be the final slide, and of course, after I'm open to any question. First thing, it's still an ongoing work. So for the moment, I do not have the ability to keep on working on that task, but I found it, a bit deceptive, I can say so. And I'm quite actively on the lookout for collaborations from both the biological side, so meaning that I would need tumoral profiles to deconvolve to see and evaluate the performance on realistic data sets of decovert, and also on the statistical side to help addressing some of the issues that I identified, like, for example, proving the uniqueness of the maximum likelihood solution or the identifiability. Second thing, which is even more generalistic, I think that it's really important to promote scientific activities and collaboration across teams. And one way, or there are plenty of ways to do so. So far, what have been organized is a symposium, sort of mini symposium showcasing the most popular deconvolution approaches so far. And I co-organized one in Jobim, so in Toulouse, in France, last year. But we could also consider more international symposium, because it was rather national, I could say. We could also consider academic exchanges between students and teams. We could consider also the development of workshops, and one way to get some funding is to organize, for example, an Elixir hackathon or a MBL workshop of 1 week long. We could also consider conferences to showcase and consortium publications to showcase and promote the best reproducing practices and the best decommission practices depending on your biological context. So here ends my decommission speech. I'm really open to collaboration, to your questions, to promote this kind of activity, which I found really interesting and meaningful for the world community.
Thank you, Doctor, for the great presentation. We are so lucky to, just be watching this as you provided this information for us. And I know there's a lot of information for some people to catch on. So I'm gonna give them a little bit of time. And I'm gonna ask you some of my questions along with Mona's question. She asked,
What I also ask you for question. Okay, right.
she asked, "Do you have any publication on his great work? So if it would be possible for you to share with us."
Yeah, so I have both a GitHub repository where we certainly need to update it, but if you want to run some simplistic frameworks and to run or estimate your server ratios, it's available as a GitHub repository. And I have also an archive paper actually deposited on the AL archive, so it's like the repository for the French researcher community. I can share you both of these things afterward, and you can provide them with the community. As I said, I'm really open to collaboration, and for me, since it's more or less published, even as an archive repository, you can use it, you can try, you can reach me out. It would be my pleasure.
Great. When you're talking about the collaboration and stuff, I want to approach this as someone who's from the biological side, and I know you have a background in biotechnology. As I was watching your presentation and I was listening, there was some packages you said you were working with. Can you just give a brief information about these packages and how can they make it useful for them?
So I think you can go back from one slide or two.
Yeah.
Okay, again. I think there's someone which is missing. Okay, no, go on the other way, sorry. On the other way, sorry. Yeah, no. Yeah, this one. So you can, I can share you also the link for that. And I would say, the main coordinator now is Francesca Finotello, and she has developed with her team. So it's mostly composed of PhD students, but maybe at some point she will also hire a postdoc. And so the idea of this blend of 7 packages is to streamline the deconvolution of bulk omics by providing curated and a unified interface to a variety of the most popular gold standard deconvolution approaches. Like, for example, in immune deconv, so for deconvolving bulk transcriptomics samples, without relying on single-cell figurines, and it's often mentioned as the first generation of deconvolution approaches. CyberSort, I think it's one of the most performing and the most popular in the field, will be implemented within it. So in these packages, what you will need to provide with is, at the very least, your bulk transcriptomic profile, of course, at the cons level. This is another thing that is really important to consider. There's a variety of benchmarks that showcase that all the pre-processing and normalization, like, for example, log2 transformation or TPN normalization or quantum normalization, usually they decrease the performance of deconvolution algorithms. So you really need to have access to the raw cons, the purest raw cons as possible. and not even normalized for library depth sequences. There is a variety of technical and theoretical explanation that try to explain this fact, but you really need the raw form. This is where you're going to perform the best. Secondly, in most of these deconvolutional approaches, like for example CyberSort, they come up with their own reference profile, and I think it's called LM22, for example, for CyberSort. So it characterizes the profiles, the transcriptomic profiles of 22 immune cells that are quite likely to be found in blood samples. But I should warn you beforehand, LM22 has been designed to be quite flexible and versatile, if I can so, and it not only incorporates immune cells that are available in the blood, but also ones that are only available in tissues. For example, it incorporates both monocytes and macrophages. While in tissues, you are expecting to only come across macrophages, and in blood, only to come across with monocytes. So it could be interesting, since they are really strongly correlated, these two elements, to discard and to completely remove the colon associated with the macrophages if you know that you are going to extract information from blood samples. Otherwise, your results are going to be systematically biased. So it's quite important to curate the reference matrices that you provide to your deconvolution framework.
Great, thank you. There's another question. Why do the normalization methods decrease performance of the convolution method?
I would say the most recurrent hypothesis from most statisticians is that when you're running and performing transformation or normalization, you break the linear assumption, which is fundamental in your linear deconvolution framework. For example, in log2 transformation, when you apply log2 transformation, you're going to really lose the linearity. And usually, you should know that, of course, they are really interesting with all these preprocessing operations to better delineate cell types, or to better delineate the biological signal. But most of the preprocessing operations, they actually also will remove the technical noise, but they also remove part of the biological signal.
Great. And another one, someone asked if this method, because I think they're asking this because you were talking about immune cells. So they asked if is this only applicable on immune diseases, or just you can apply it?
No. It can really be extended to any kind of disease. The true factor which is the most important here is the quality of the reference profiles that you're going to provide to a deconvolution framework. So, for example, if you consider to deconvolve monkey tissues, so not human, if you are going to provide a human-based signature for your cell profiles, you are certainly going to observe worse performance compared with you provide directly monkey cell-specific profiles. So here it's for a species, but it's the same applies for a given tissue or a given tumoral subtype. The most important is that your cell-specific expression profiles is the closest to the true biological reality. So do not include irrelevant cell profiles, but do include all the cell types that you may consider present and significantly present in the bulk profile that you want to deconvolve.
Great. There's another question. Have you performed any benchmarking survey comparing, preferably compared to a single cell experiment?
What do you mean exactly by this? Because with single cell, you don't really need to apply deconvolution because you already have access to the more or less to the facts information or to the single cell level information. I'm not sure if I get it well, the question. Maybe if I got it well. So usually the benchmarking, when you benchmark a set of deconvolution algorithms, the gold standard is not single cell, but facts or over cytometry-based approaches, because they are relying on surface protein markers, they are supposed to be the most unequivocal to characterize cell abundances. But I'm
Um,
not sure if it
I
covers.
think they mean if you have compared the results with to validate, with the single cell experiment.
Mhm. So the quick answer for that is no. Usually, as I said again, the benchmarking of deconventional approaches is usually done by doing two things: by implementing and using and generating completely simulated data set in which you know the ground truth because you simulate them, or if you are going to perform on real data sets by comparing the proportions that you retrieve with your deconventional algorithm with respect to others and with respect to gold standards, which are assumed to be the facts ones. Maybe there are other cytometry approaches that have been developed, like CyTOF, that are more performing, that are even more performing compared with facts, but I do not know them.
Okay, thank you. I think I don't think there's another question. So, oh, okay. Is your approach applicable for a microarray and even mass spectrometry?
So microarrays, I do think so. The distribution is a bit different here because it's more or less Gaussian distributed actually. So it may be even easier to apply the covert on this kind of information or this kind of sales. The other issue with microarrays is usually that it's truncated on both sides. So on the left hand side and on the right hand side, like the most extreme values. But I do think that should be feasible. I should certainly need to adjust the density distribution to fit my bulk profiles. But yeah, that should be feasible. And actually, for example, if I'm considering LM22, so the signature matrix used for cybersort, they generate it by mixing up microarray and RNA-seq derived expression. For mass spectrometry, I'm not sure if, but I'm not a biologist from background, so I may lose the point here. So, what's the general purpose of mass spectrometry?
Um, Let me see if they wanna. Mizak, do you want to open your mic and ask your question yourself? To quantify the proteomics profile.
To quantify proteomics. Um, I think there have been like two or three, or maybe a little more, deconvolution approaches that have been developed to deconvolve proteomic samples. However, I do not really know them. I know also that there have been deconvolutional approaches, at least two, that try to incorporate both transcriptomic and proteomics to even improve the performance of deconvolution. However, so far, Decorat could not be used to and is not tailored for deconvolving proteomic mass spectrometry samples.
Thank you. I think we got them all, and that was great. I hope that we can collaborate on stuff more than this. And I'm pretty sure there are people really excited about this kind of research, and they want to collaborate. So it would be okay if they contact you with like LinkedIn and other ways. Yeah.
Yeah, so I think I provide you with both my LinkedIn profile and if I remember well, my personal email address, which is certainly more stable compared with respect to my professional email address. So yeah, feel free to reach out on using either my personal email address or LinkedIn. I think on my LinkedIn, since I do not have the premium, you can send me a link only or message me directly only if you first reach me out or ask me for an invitation. But yeah.
Great. Thank you again, and I'm sorry about the misfortunate at the start of the.
No worries.
Really sorry. I hope that we can do something like this again. And thank you for the time you took and for the great presentation.
بعد این 0 و 1 هم که
Yeah,
هست میبینی
you're
گل
welcome.
زدنه اینو این
Thank
دفعه.
you and have a good day.
Yeah, thank you very much. See you. I guess it's the night for you now.
Yeah.
یه هم یه کوچولو
What time is it actually in Iran now?
بعد
It's
اتفاقا
9 p.m. So like 21, yeah.
Oh, I'm quite impressed that most of the people they stay up to the end of the presentation. Even
Yeah,
the
they were really excited to hear you talking about this research as I. So thank you again.
Yeah, thank you very much.
Have a great day.
Yeah,
واسه
see you.
الان واسه پیر ریویوش هم باید اینا باشه. تو داری رو پیر ریویو رو میگی.
خب دوستان خسته نباشید. من تشکر میکنم از همتون که تا این ساعت بودید و استفاده کردید از ارائه مثل خودم. میدونم که ممکنه برای خیلیاتون قسمت‌های سخت باشه و نفهمیده باشید. اصلا نگران نباشید. سعی کنید که به اندازه خودتون از هر ارائه‌ای برداشت کنید و همون میگم شما 5 تا 6 تا کیورد هم بردارید و اینا رو برید دنبالش سرچ کنید، بگردید. با خود دکتر هم که میتونید در آینده در تماس باشید و از طریق همون لینکدینشون یا ایمیلشون و اینا که حالا هستش در اختیار همه، میتونید سوالاتی که دارید رو بپرسید و ما هم همیشه در خدمت شما هستیم و تا جایی که بتونیم سعی میکنیم که برنامه‌های خوبی رو براتون تدارک ببینیم. خیلی ممنونم از همگی. اگر سوالی، اگر چیزی نیست لطف کنید فقط من برای بار آخر هم اینو بگم که این لینکی که من اینجا گذاشتم پین کردم برای اینه که ما بتونیم برنامه‌های بهتری برای شما تدارک ببینیم. پس لطف کنید اگر یه چند دقیقه وقت بذارید و این فرم رو پر بکنید خیلی خوشحال میشیم و برای خودتون هم خیلی خوب خواهد بود. بازم تشکر میکنم از دکتر و خانم پاتوقی عزیز که بودن اینجا و سوالشون پرسیدن. ممنونم از همتون. اگر که چیزی نیست من شب بخیر میگم و امیدوارم که شب خوبی رو داشته باشید. وقتتون بخیر.
بعد انگلیسی یا فارسی میگی؟
انگلیسی ترجیحاً دیگه همیشه چیزامون انگلیسیه.
خب الان همین مشکله دیگه. مثلا 17 اپریل 16 اپریل تبدیل
دیگه ببین وقتی زیر شاخه انجمن بین‌المللی باشه از این بدبختی هم داری. ببین به نظرت بد میشه اگه این بنر باشه مثلا پوستر نباشه افقی باشه به جای اینکه عمودی باشه؟
آخه تو این استا میذاریم جا نمیشه میدونی؟ اینه واسه یا جای دیگه اون ابعاد 10 در 8 80 در 19 20 نه 13 در 5 13 در 20 همچین چیزی یادم میره. اون همه جا اوکیه تقریبا. رو سایت میشه رو این ور اون ور. میخوای یک چیز کوچولو میزنیم خب بعد چیز کنیم بفرستم تو اون گروهه بعد یا خواستن جلسه بزنیم با اون دوتا.
شدن رو میشه اوکی. ببین من میتونم به فاطمه هم بگم که اوکی کنتشا ولی نمیدونم میتونه برسونه تا هفته بعد یا نه. البته کار خاصی الان نداره ولی کلا این ذره جواب میده. بعد نمیدونم نظر خودت چیه؟
من فکرم اینه که این پسته پیشه با پسته یه چیزی داشته باشه. یه
کار دیگه هم میتونیم بکنیم فاطمه.
چی؟
این پوستر رو خب همون پوستر رو برداریم اون چیزای اضافهش رو جدا کنیم اون نوشته ها و اینا رو همه رو ریموف کنیم خب بعد بلورش کنیم یه ذره محو بشه روش نوشته بیاریم دوباره
بعد
روش نوشته ها و چطور
اذیت میکنه یا نه؟ مثلا با گوشی همون و یه سری چیزایی کار کنه خوب در نمیاد، بیشتر تو آیفون خیلی خوب در میاد. دیدی مثلا بالا بیوتک نتورک بلر میکنه چقدر خوبه؟ و حالا خودت بلر کن گوشی هم چقدر چیز میشه.
ببین اینو اوکی، خب چون اینا رو میخوایم با چیز کار کنیم، با گوشی کار کنیم، اینجوری میشه. اگه مثلا با ایلاستریتر مثلا کار میکردیم اینجوری در نمیومد.
یه چیز کوچولو من میذارم میفرستم تهش اینه که بعد تو فیگما میذارم خودشون میاد ادیت بزنن، یه کاری میشه دیگه جزئی.
آره اینم میشه، اینم میشه. آره میتونیم امتحان کنیم و انشالله که جواب بدیم. خب بعد به نظرت مثلا این ریویو پریویو ها رو باز هم مثلا ریویو با پریویو فرق کنه؟
یه چیزیش باید فرق کنه
یا
دیگه.